What's up, Ninja Nerds? In this video today, we're going to be talking about the cell nucleus. Before we get started though, hit that like button, comment down in the comment section, please subscribe if you guys like this video. If it helps you, if it makes sense, please do that. It helps us to continue to keep making free videos for your enjoyment. Also, we got some awesome notes, illustrations, check them out in the description box below, take you to our website. Great tool to utilize to help you guys to understand this topic and follow along with me. Without further ado, let's talk about the cell nucleus. So, the nucleus, we got to talk about is the structure and the function. So what I really want to do is I want to cover the components of the actual nuclear structure. And it's really kind of two things. You see here we have a cell, and then again, within the cell you have your intracellular fluid, and then here you have your extracellular fluid. We already talked about the cell membrane, how it's separating those two. Right? We went over the membrane lipids, the membrane proteins, the cholesterol, the glycocalyx structure, all that stuff. Well, here, I want you to think about this structure here surrounding the inner side. So this right here is your nucleus. This whole thing right here is your nucleus. But there's a structure that's surrounding the inner components of the nucleus. You see this? This is called your nuclear envelope. And we'll go over all the different components of the nuclear envelope. But it's really cool. There's three components of it, and we'll talk about that structure here. The next one here is inside of the nucleus, there is the nuclear content, and we'll go over in great detail what the nuclear content is. It's primarily going to be made up of two things. One is the nucleolus and one is the chromatin, and we'll go over that in more detail later. But let's actually take some time now. We know the two structural components of the nucleus is the nuclear envelope, and then the next one is the nuclear content. Let's dive into each one. Nuclear envelope. If we were to take this nuclear envelope here, this little son of a gun right here, and zoom in on it in great detail, this is what we get. This is our nuclear envelope. And within the nuclear envelope, you have this part here. And this is going to be the membrane. And there's two components of this membrane. And I'm not kidding, it's very straightforward. This is the outer membrane. Oh man, super complicated, isn't it? So first one is the outer membrane. And again, this outer membrane is made up of phospholipids. It's made up of phospholipids. So you're going to see the phosphate groups, you're going to see the fatty acid tails, you may even have some cholesterol within that. The next thing here is you're going to have the inner membrane. This is the inner membrane. And this is going to be another really important component as well. And as you see here, the inner membrane contains, again, this phospholipid bilayer as well. So here's your phosphate groups, here's the fatty acid tails, and then there's another important component here, which is called your lamina. So again, outer membrane is made up of a phospholipid layer, and then this one contains a phospholipid layer and a lamina layer, which is made up of these proteins called lamins. Very important proteins, okay? And then the last thing is you have these like orange proteins which are kind of plugged in and span, they're integral proteins or transmembrane proteins that span the entire nuclear envelope. These are called nuclear pores. So these are called nuclear pores. So again, let's go ahead and quickly recap up to this point. What have we covered? Nuclear envelope consists of outer membrane, which is a phospholipid layer. Inner membrane, which has another phospholipid layer, but this green layer here is called the lamina layer, made up of lamin proteins. And then lastly here, we have the nuclear pores. And the nuclear pores are going to be the proteins that span the entire nuclear envelope. Nuclear content. So now we have the nuclear envelope, which is the outer membrane, inner membrane, nuclear pore. Inside is what? All the juiciness, right? In this part here, this red structure here, this is called the nucleolus. So it's called the nucleolus. All right, so the nucleolus. That's this component here. And then surrounding kind of all these like squiggly lines around the edge of it is called the chromatin. And we will talk next about all of these different things. So now what we've done is we've completely covered everything with the structure of the nucleus. We've covered the nuclear envelope, talking about outer, inner, and nuclear pore. We then talked about nuclear content, which is the nucleolus and the chromatin.
All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells: The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made.
Now, let's dig into a little bit more detail on the functions of the nuclear envelope and then functions of the nuclear content. All right, my friends, so now we get to talk about the functions of the actual nuclear envelope, okay? So when we talk about this, we're going to go through each kind of step in a little bit more detail so we know that it's made up of the outer membrane, the inner membrane, and we know it's also made up of the, again, nuclear pores. Is there some specific functions to that? Yes. So outer membrane is actually really cool because one of the cool things is, as you can see here, it's contiguous with the endoplasmic reticulum. Very specific one though is it's very continuous with one of the structures called the rough endoplasmic reticulum. And I love this because it makes so much sense why it's continuous. So here is our chromatin. What you'll learn later is that chromatin is basically DNA, okay? And DNA will undergo this process called transcription, where you'll take DNA and convert it into mRNA. So then we'll make this into what's called mRNA. And this is a process called transcription. mRNA loves to bind to what? The ribosomes. So we call this structure the rough endoplasmic reticulum because it's this kind of like these continuous tubules that are continuous with the nuclear envelope, particularly the outer membrane. And what it has on it is it has ribosomes. And what happens is this ribosomes, when they get read by the when the mRNA gets read by the ribosomes, what it does is it pushes into the rough endoplasmic reticulum a protein. So it pushes into the rough endoplasmic reticulum a protein. Isn't that cool? And then what happens is while it's in the rough endoplasmic reticulum, it undergoes some specific modifications. And then when it's done being modified in the rough endoplasmic reticulum, it takes and says, okay, I'm done modifying you. Let me go ahead and bud a piece of you off into this vesicle here. And then what I'm going to do is I'm going to break you off and then send you somewhere else to get further modified. And that's one of the really cool concepts of the outer membrane. So what I want you to remember is the outer membrane of the nuclear envelope is contiguous with the rough endoplasmic reticulum. And why does it have to be in such close proximity? Because the DNA in the nucleus will get transcribed into mRNA. mRNA will get translated on ribosomes on the rough ER, which is very contiguous with the outer membrane, and then get utilized to make proteins. So protein synthesis get packaged and then sent to another structure called the Golgi, which we'll talk about later. That's one really, really cool concept of the outer membrane. The second thing is the inner membrane. What is the inner membrane good for? Don't say absolutely nothing. All right, the inner membrane is really, really cool. So here we have the outer membrane, which was contiguous with the rough endoplasmic reticulum, which is an endoplasmic reticulum studded with ribosomes. DNA, mRNA, mRNA to the ribosomes, gets translated into proteins, pushed into the rough ER, and then modified and then sent to the Golgi. The inner membrane, a little bit different. So one of the big things, the inner membrane has this protein here called lamins. All right, so it's called the lamina, but lamina is basically made up of lamin proteins. And these lamin proteins have many, many different functions. What are some of them? they're involved in taking this here. This is DNA, right? DNA is basically going to be right here. I'm just kind of zooming in on it. When DNA undergoes a very specific process where it makes more DNA, what is that called? DNA replication. This is important because in order for us to replicate DNA, it allows for us to take one cell and make what? 2 perfectly identical cells via mitosis. So it's involved in the cell cycle in that particular way. It also, and then again, lamins is basically the thing that's helping and assisting in this particular process. So let's actually draw a green arrow or a green plus sign to recognize that the lamin proteins are completely involved in this process. They're also involved in these 2 other processes, which is what? Taking DNA and converting it into RNA. You know what this is called? This is called transcription. This is called transcription. And there's so many different types of RNA. There is what? mRNA, which is the most important one, which will get translated by ribosomes. There's tRNA, which is important in protein synthesis. There's rRNA, which is also important in the ribosomal formation. So all of this is kind of helped with lamin proteins, which are on the inner membrane. And the last thing is they organize the DNA and condense the DNA. And we'll talk about this later. So you see how all of this kind of DNA here is kind of condensed down near the edge by the lamins? The lamins help with that process. They organize and then compact and condense the DNA as well. So these are the 3 particular functions of the inner membrane, with most specifically involving the lamin proteins. They're involved in DNA replication, DNA transcription, and organization of the DNA inside of the nucleus. Let's now come down to the last component of the nuclear envelope, which is the nuclear pores. And we already kind of indirectly covered what they do, and it's really, really cool. So the nuclear pores are great because inside of the nuclear envelope is the nuclear content, the chromatin, which is DNA and proteins, and the nucleolus. In order to make DNA, what do you need? You need ions, you need nucleotides, you may need some proteins, right? So if I want to transport things into the nucleus, such as ions, or such as amino acids, or such as nucleotides, I have to use these nuclear pores to import these particular small molecules in. And so these nuclear pores can allow for import. So maybe there's one example of importing specific types of molecules passively. This would be a passive example. They don't require any type of ATP to perform this. So there's no ATP required. But let's say that I want to move out large molecules, such as RNA, right? RNA. In order for me to get this big molecule RNA out, I may need to utilize ATP, so that's an active transport. And this may be involved in exporting the material out of the nucleus. And so if I move this RNA out, let's say that this specific RNA is mRNA, and I give you tRNA. You know what these generally do? These 2 go and combine with what's called ribosomes. Ribosomes. You know ribosomes, these are very, very special structures that we already talked about that are present where? On the rough endoplasmic reticulum, or they can be free ribosomes. But generally what happens is the mRNA will interact here, and then the tRNA will interact here, and this will help us to synthesize proteins. So this will help us to synthesize proteins. And some of these proteins maybe need to be sent back in to the nucleus. Very, very large proteins. And so let's say that some of these proteins that I basically have synthesized, I want to send back into the nucleus. Well, then in order for me to be able to get these proteins back into the nucleus, because maybe it's involved in some type of particular process, I may need to import them again. But if they're really, really large, so if I have to import large molecules, right? Import small molecules, it could be passive. Importing large molecules may require ATP to do this, so it's active. One of the cool reasons why this is important is because, you know, inside of the actual nucleus, there's something called the nucleolus. There's this structure here called the nucleolus, and we'll talk about this in just a little bit, but the nucleolus is really, really important because what it contains is, it contains something called rRNA, and it contains something called ribonucleic subunits. And I think that's super cool because we actually synthesize proteins, like ribosomal subunits, send them back into the nucleus, and then what happens is this actual DNA will make RNA. Guess what's the other RNA that it makes? rRNA. And if we combine that with the ribosomal subunits, this will then kind of go into this area here called the nucleolus. And then it'll help to combine it and make true ribosomes. And then from there, we'll send out these large proteins out of the nucleus, out into the cytosol to bind onto the rough ER so that it can perform more of the actual translation process. So that's a really, really cool concept. And again, we'll recap this in a little bit. But the basic component of the nuclear pores is to allow for the export of things from the nucleus out into the cytosol, or to allow for things to come from the cytosol into the nucleus. Big thing to take away from this is that if it's large molecules, it requires ATP to import them or export them. That's an active process. And if it's small molecules like ions or small amino acids or small proteins or nucleotides that are needed and they're being imported, then that is actually going to require no ATP and it's a passive process. Okay. At this point, we have covered now the functions of the nuclear envelope, that being the outer, inner membrane, and the nuclear pores. Let's now move on to the functions of the nuclear content. In other words, going over the nucleolus a little bit more, and then going over the chromatin. All right, my friends, so now we're going to talk about the functions of the nuclear content. So first thing I want to talk about is, again, when we talk about nuclear content, we said that it's made up of chromatin and the nucleolus. Just like when we said nuclear envelope was outer membrane, inner membrane, nuclear pores. Outer membrane, continuous with the rough ER, really involved in that kind of process where DNA in the nucleus gets converted into mRNA, combines with the ribosomes on the rough ER, and then helps to make proteins. Inner membrane has lamin proteins, which are involved in helping the process of DNA replication, DNA transcription, and organizing the DNA. And the nuclear pores allow for things that are molecules or such to move in and out of the nucleus. If it's large, charged molecules, big proteins, big nucleic acids, those require ATP to be imported or exported. If it's small ions, amino acids, small proteins, and nucleotides, those can be imported or exported without ATP. That's a passive process. Okay. Next thing is chromatin. Chromatin is really just made up of two particular things. So if we were to actually take this chromatin inside of the nucleus, it's actually going to be made up of one thing which is called histone proteins. So good old histone proteins. And the big thing I want you to know about histone proteins is that they are positively charged. And there technically is 8 of these son of a guns. 8 of these different types of histone proteins. With the histone proteins, because they're positively charged, they're going to love to interact with things that are somewhat negatively charged. And what do you think is negatively charged, my friends? DNA. So DNA is the other component here of chromatin, and DNA is this double helix, and it's actually negatively charged. Now, that is the two components of what? The two components of chromatin. So one is DNA, and the other one is histone proteins. These make up most of it. There's other small percentages of things that are involved in chromatin, but these are the two primary things. Now, when we talk about chromatin, really, what is the function of it? So let's actually kind of dive into a little bit. Let's talk about what are the functions of DNA, and then really what are the job of the histone proteins, because we know that that's what makes up chromatin. Well, DNA is really cool in the sense that I already know that I can take DNA and use specific DNA polymerases to take this DNA and make more DNA. So if I want to take DNA and make more DNA, which will help me in the process of replicating cells, so taking one cell and then making 2 cells, this is an important process via DNA replication. So that's one of the things that I could do with DNA, is I could replicate it, which is important in making more cells. What is this called when I take one cell and make 2 identical cells? This is the process called mitosis. And this involves so many cells of our body. Another concept is, what if I take the DNA, I take a piece of it, a gene, a very specific portion of it, and I read it and I transcribe it and make RNA. Specifically, with these examples, is mRNA. But there is other RNAs, right? I can make mRNA. I could also make another RNA called tRNA. That's another example. And I can make another RNA, which is important in ribosomes, which is called rRNA. What is this process called when I take DNA and make RNA? This is called transcription. Why is transcription important? Transcription allows for you to take DNA to make RNA. RNA, which is mRNA, tRNA, rRNA, are all involved in the process of reading mRNA and making it into something called proteins. And proteins are super, super important because they're basically the things that keep the cells going. They play a role in structure, they play a role in function. And so very, very important process there. So DNA is really cool in the sense that, again, you have the ability to replicate it or transcribe it to make very specific many types of proteins that play a huge role in our cells being able to survive. The second thing is the histone proteins. What do these things really do? What do these little guys do? It's actually really cool. One of the concepts here is, let's say here we have some histone proteins, right? So it's an octamer. There's 8 of these histone proteins kind of locked in here. So there's 8 of these histone proteins that are kind of locked in here. What happens is DNA will wrap around this actual histone proteins twice. Isn't that cool? So they'll wrap around these histone proteins twice, and what that does is that allows for these kinds of DNA to get condensed. So what we call this is really whenever these are kind of wrapped around, the DNA is wrapped around the histone proteins, we call this a nucleosome. Nucleosome. And what happens is you can take DNA. You know how long DNA is? It's 2 meters long. Imagine trying to fit 2 meters of this stringy DNA into a nucleus. You can't do that normally. So the histone proteins will condense the DNA. Look how it condenses the DNA to make it fit into the actual nucleus. And that's important because we need the DNA to condense in order for us to undergo the process of mitosis. Because we don't want to have DNA being super loose and stringy, because then it can actually cause lots of breaks and damage to the DNA. And that's why, you know what one of the best examples of condensing of the DNA is? You know what an example of that is, especially during mitosis? We take the DNA and we compact it into this thing called a chromosome. That's all DNA in there, but a chromosome is going to be one of the best examples of condensed DNA. DNA wrapped around histone proteins to really allow for it to fit properly in the nucleus and allow for it to easily be passed on from one cell to the next without breaking strands or messing up the DNA. Really, really cool. Another function of the histone proteins is that they play a role in regulating, they regulate genes. They play a role in gene expression. So imagine here, there's a particular gene right here that I want to read. And I want to read it, and I want to be able to make something called mRNA from it. That's really what I want to do. I want to be able to read it and make mRNA, so transcription. These histone proteins, if you put certain things on them, it will help them to maybe tighten the DNA up or loosen the DNA. So in other words, if the DNA is tight, can I actually read that kind of gene? No. If the DNA is looser, will I be able to easily read that DNA and make an RNA? Yes. So here's the thing. If I were to add on a methyl group, so I'm going to add on what's called a methyl group. I'm going to do something called methylation. Methylation is the stop sign to the gene. Now look what I'm going to do. I'm going to condense this DNA to where I can't read the gene. And what this is going to do is, if I have this methyl group here, so imagine here I add a methyl group onto, I'm just going to represent it with this dot here. Here's the methyl group on these histone proteins. They will condense the DNA. So when they condense the DNA, we actually say it's really hard to be able to provide transcription. So we will inhibit transcription. You will not make any RNA. This type of DNA that's condensed, we give it a very specific name. Right? We call this heterochromatin. Heterochromatin. I want you to remember highly condensed for heterochromatin. Highly condensed. And this is usually due to histone methylation. In the other situation, if I add a acetyl group, all right, so usually this is kind of like this kind of structure here. I'm adding some type of carboxy group with like a methyl group on. This is called acetylation. Acetylation. Okay, so I'm adding some type of carboxy group with like a methyl group on it. So here I'm going to draw this one like this. I have these acetyl groups that are added on to the histone proteins. Believe it or not, it allows the histone proteins to then separate the DNA strands and open up the genes. So now this gene is super opened up. And since it's opened up, now the actual gene expression is in play. So this will stimulate transcription. And if I stimulate transcription, guess what I'm going to make from this DNA? I'm going to make RNA. And that is a really cool concept. So it doesn't condense the DNA. Instead, what does it do? It actually loosens, if you will, the DNA. You know what we call that when you loosen the DNA? It's called euchromatin. So it's called euchromatin. And the way that I remember this one is expressed DNA. So highly condensed DNA, expressed DNA. Methylation shuts down transcription. Acetylation stimulates transcription. Methylation will cause heterochromatin. Acetylation will cause euchromatin. That's the big things that I want you guys to take away from the chromatin function. Let's now talk about the next aspect here, which is the nucleolus. So the nucleolus is the other component. We talked about chromatin being the DNA and the histone proteins and what they do: DNA replication, transcription. We talked about chromatin for the condensation. We also had histones for the condensation of DNA, and then we also talked about it for gene expression. The nucleolus is really cool. And what I want you to remember, big thing is, nucleolus is the site of rRNA and ribosomal synthesis. Let me explain. Okay, from this DNA, I'm going to make 2 RNA molecules, or we'll say 3. We're going to make RNA, okay? One of these is called rRNA, and that goes over here to this nucleolus site. We'll also make tRNA and mRNA, and these will go out of the nucleus via the nuclear pores. So here I'm going to have mRNA, and I'm going to have tRNA. These will then go and actually interact with a ribosome. So here's going to be a ribosome. And ribosomes can be free, meaning they're circulating in the cytosol, or they can be bound to the rough endoplasmic reticulum. The mRNA will bind with the ribosome, and the tRNA, which we'll talk about later, will carry a very specific type of amino acid. And from this, they will synthesize what? Proteins. So from this, the whole goal is to synthesize proteins with the mRNA and the tRNA. Okay? So here's going to be our protein. One thing I left out, though, is that there's one more RNA. So this is tRNA, which carries the amino acid, the codon, or the anticodon. mRNA carries the specific code, the codon. But there's another molecule in the actual ribosome called rRNA. The nucleolus is actually what synthesizes the ribosomal subunit and the rRNA. But how does it do that? We're almost there. So let's say we're making these proteins, and just for the simplicity's sake, let's say that it's ribosomal proteins. So let's actually make this red. Here's my ribosomal proteins, or my subunits, okay? Where do these have to go? Well, where the rRNA is. Where's the rRNA? It's in the nucleolus. So what I'm going to do is I'm going to take these ribosomal subunits and I'm going to bring it in. And I'm going to combine the rRNA and the, what's this right here? Let's actually just show them in vivo here. Here's going to be our ribosomal subunits. I'm going to import them into the nucleus, combine these two. And when I combine these two, I'm going to make ribosomes. And guess what I'm going to do? Now that I have these two, I'm going to combine them together, and I'm going to ship out of this nucleus via exporting, via the nuclear pore. I'm going to send out my ribosomes. Here's my ribosomal subunit. Here's my ribosomal subunit. And guess what's kind of incorporated into the structure? The rRNA. That's a really cool thing. And then that will then kind of go and help you to make more of these, which will help you to make more proteins. So really the nucleolus is really the site of taking R. So if we were to really take this nucleolus and zoom in on what happens, here's my rRNA. Here is my ribosomal subunits. The rRNA comes from the DNA. The ribosomal subunits comes from the protein that was made via mRNA, tRNA, and a prior ribosome that had RNA and proteins in it. And then from this, I will synthesize more of these. So it just keeps manufacturing more of these ribosomes that will either become free ribosomes or will become ribosomes that bind onto the rough endoplasmic reticulum. So the nucleolus is the site of ribosomal synthesis, where rRNA comes from the DNA, and the DNA makes mRNA and tRNA, which helps to undertake translation, making more ribosomal subunits, sends it back into the nucleus and combines with the RNA, makes ribosomes, and sends them out via export. Isn't that cool? I think it's pretty cool. But that is the concept there for the nucleolus. And that finishes the video on the nucleus structure and function. I hope it made sense. I hope that you guys liked it. As always, until next time.